Okay, so let's leave music and poetry and come back to the rather prosaic world of bacterial structures. So we've done the different types of pili, and now we're going to finish up our consideration of the external structures of bacteria with flagella. And flagella can be present in one per cell or several. So if you've got one flagellum at one extremity of the bacillus, then these cells are called monotricious. They have one hair. If you have several at the same extremity, this is called lophotricious. And if you've got flagella all around the bacterium, then this is called peritricious. Flagella are present in gram positives and gram negatives, perhaps a bit more frequent in gram negatives, and they're only expressed by bacilli. Okay, I don't think any cocci have flagella. So that's how they're arranged around the bacterial cell. If you look a little bit at the molecular structure, then in gram-negative bacteria, there's a big protein complex that anchors the flagellum in the plasma membrane, crosses the peptidoglycan layer, another anchor in the outer membrane, and this forms a kind of axle. And at the top you have a hook made of protein to induce a bit of curvature here, so that the flagellum isn't pointing out perpendicular to the surface of the cell. And then a long filament made up of thousands of copies of the same protein, flagellin. So this filament will be 15 to 20 micrometers in length. So a bacterial cell is maybe 1 to 10 microns in length. So the whole filament is maybe 3, 4, 5 times longer than the bacterial cell itself. Okay, so if it's like I had a whip here, it would be like going all the way over to the other corner of the room. That's a relative size to the bacterial cell. Now, the flagella protein, the flagellin, this can be variable between strains of the same bacterium, and that's why it's useful for identification. So the flagellum is also known as the H antigen in bacterial serotyping. So H is for heat sensitive because unlike the O antigens, LPS, and the K antigens, the capsule, the H antigen, the flagellum, is made out of protein. So when you heat the protein, the antigens are denatured, antibodies don't react anymore. So if you see something like this, E. coli H7O157, H7 is the serotype of the flagellum. And O, that's the serotype of the LPS. Okay. Right, so that's the structure. Their function is to move bacteria around in a liquid medium. And the way it works is that this whole axle can rotate. So the flagellar motion isn't like waving around like this. It's not that. It's kind of rotating. And this is what generates the force to push the bacterium around. So to generate this movement, bacteria have to expend energy, right? And the energy source for this is don't be sorry, be on time. Okay, so the energy source for this is the proton gradient, the proton motive force. So you have a more acidic pH in the periplasm than in the cytoplasm. So that means there's a proton gradient. So if protons move from the periplasmic face to the cytoplasmic face, this is energetically favorable. It's exergonic. And this energy can be used or can be harnessed to perform some kind of work. And what happens for the flagellum is that as the protons are transported from the periplasm to the cytoplasmic phase, it's going to result in turning around the rotor here. The axle starts to spin around as the protons are transported. Now down here you have what's called the switch protein. So depending on the activation of the switch protein, the flagellum can rotate in a clockwise direction or an anti-clockwise direction. And that's really going to be, So just before I move on to that, so how does the flagellum get elongated, how is it produced by the cell? So the basal structure is hollow, this axle here, and the subunits of flagellin can be transported up the inside of the base structure. The flagellum itself is hollow, so that all the new flagellin subunits travel all the way up the inside of the filament, and they're added to the tip. And then as you add more and more flagellin subunits, then the flagellum gets longer and longer. So does this remind you of anything? Like among the pili, yeah. Maybe, yes. What was that? Actin, myosin, not really prokaryote protein, but more recently, like this week, what did we see about the types of pili, the way they're formed? So if you remember, the curly and the CU and the type 5 pili, they're all anchored in the outer membrane, so it's not like that. And then type 3 and type 4, they both have a big structure that crosses both membranes. And the question is, which one?
It's type 4.
It's, unlucky. Type 3 is type 3. So type 4, the subunits are added on at the base. That's why it can be extruded and retracted. The bacterium can control this. Type 3, they're transported up the inside of the filament and add on the tip, just the same as this. Okay? So the theory or the way people think is that the bacterial flagellum is, or in gram negatives anyway, it's a modification of a type 3 pili. Okay, because the basic structure is kind of the same. So this is a complicated molecular machine doing something quite tricky, moving the bacterium around. But bacteria didn't have to build this out of nothing. Okay, it's not like, imagine you're some kind of like person building a car in your house and you have a part from another car and say, oh yeah, I'll just stick this on here and now I just add a motor and away we go. It's kind of like that. So bacteria, probably they already had type 3 pili and they just, let's just stick a motor on the bottom. The thing's going to go round and round. It'll be great. Okay, so that's where flagella come from, or how they evolved. Okay, so just for kicks, you stick a motor on the end of at the base of your type 3 pili, and that means that it can move around. Now, moving around is great, but what bacteria need to move around for is in order to move towards things that the bacteria like, for instance, sugar, peptides, something to eat, and move away from things that bacteria don't like, which might be too acid conditions, too high concentration of something toxic. So to have some kind of chemio or chemotactic response. And this has got to work in 3 dimensions. So if you were an engineer working in Naval Group to design a submarine or something, right? And you want to move your submarine around in 3 dimensions underwater. you probably think, okay, I need a motor to move backwards and forwards, and I need some control to go left or right, and then I need a control to go up and down. Okay, and then I can move around in 3 dimensions. But bacteria, they don't have that with flagella. They just have 2 options. I can move my flagellum around like this, or the other way around. And they've got to try and move around in 3 dimensions in a controlled way just with this. So the way it works is that when the flagellum is rotating in a counterclockwise sense, then the bacterium is going to go forward in a straight line. Then when the flagellum rotates clockwise, everybody know what I mean by that? Yeah, good. Then the bacterium is going to tumble around and change direction in a random way. So they've got 2 speeds. They've got forwards and random, okay, or 2 modes, if you like. So what they can do is when they switch around between these modes, then that's going to help them control where they go. And that's true for monotricious flagellated bacteria. Okay, one flagellum is rotating around in one way or the other way. And it's also true for peritricious flagellated bacteria, because all the flagella on the bacterial cell will all be rotating in the same sense, in the same kind of orientation. So all of the flagella will either be rotating counterclockwise or clockwise. And the same rule applies. So if they're all going in a counterclockwise sense, then this bacterium is going to swim forwards. If they're all rotating clockwise, then it's going to change direction randomly. Okay, so just to illustrate this, now imagine we've got a little flagellated bacterium which is hanging around, and it detects the presence of a source of glucose over here. There will be a concentration gradient with the glucose at a high concentration here and a low concentration the further that we get away. So the bacterium is going to start around just rotating randomly with the flagellum turning in a clockwise sense. And then after a random period of time, it's going to flip rotation. So the flagellum is going to start rotating counterclockwise, and the bacterium is going to start moving forwards. I don't know what happened to my arrows. They were perfect last year. So it's going to start moving forwards, and as long as the bacterium is moving up the concentration gradient, that's going to keep the flagellum rotating in the counterclockwise sense. So it's going to keep going in a straight line. But of course, what's going to happen is that it's probably going to overshoot, or it definitely will at some point. So it goes too far, but once the bacterium starts to go down the concentration gradient, then it's going to change direction again. The sense of the rotation of the flagellum is going to change, it's going to switch, and then you'll get a random change in direction, and then off it will go, start moving in a straight line again somewhere. So the bacterium is going to switch between these 2 modes, random and straight line, and it's never going to stop. It's never going to stop right in the middle here, right? It's always going to go overshooting, but if it goes too far, it will switch direction. And of course, when it switches direction, it's random. It's not choosing the right direction. So if it switches direction, it could start moving off further away. But then it will switch direction again very quickly. Okay, so if it goes in the wrong direction, it will change. If it's going in the right direction, it will keep going. So the end result is the bacteria will kind of just like swim around and keep close to this source of glucose. And that's good, right? That's what they want. They want to be near the glucose. So just with a very, very simple algorithm and one structure that's got 2 mechanisms of moving the bacterium around, then it can do something that is actually quite clever. Okay, so before we leave the external structures of the bacterium, let's try a little question, see what you remembered. And first I have to start it up on Madoc. this one. Okay, should be the question should be open now. Yes.
What is CU?
It's a type of pili. CU pili, curly pili, and type 5, they're all anchored in the outer membrane. CU stands for chaperone usher. In case you wanted to know. It's to do with the mechanism that they synthesize. Oh. But okay, got like one more vote. Everybody finished? Who's going to send in an answer? Yeah? Okay. Oh, right. So our number one answer is capsule antigens. Yes, correct. That's the K antigen. The number two answer down here, peptidoglycan, that's false. That's wrong. The second correct answer is this one, LPSO repeats. So core oligosaccharide is wrong, CUPD is wrong, peptidoglycan is wrong. Okay, why are these the wrong answers? There are two reasons why these are not correct. So for the core oligosaccharide and the peptidoglycan, these answers are wrong because they're too conserved. Okay? Peptidoglycan, it's a complex structure, but it's kind of the same in gram positives, gram negatives. There are some slight differences in the tetrapeptide repeats and the peptide bridge cross-links. But basically, it's the same thing in all bacteria, too conserved. That's also the same for the core oligosaccharide, which is very conserved within the same bacterial family or genus. So not very useful for identification. CU pili is the opposite, it's too variable. Okay, there are a lot of different types, and the same bacterium can have more than one type at the same time. So that means it's just too complicated. O repeats and the capsule antigens, they are variable between strains of the same species, but each bacterial strain will just have one K antigen or one O antigen. Okay, so that makes it useful. So that's why these two are the ones that are useful for identification. So O antigens, K antigens, and H antigens, they're the ones that help us identify bacterial strains. Okay, so now we move on to the very, very simple structures on the inside of a bacterial cell. Now, there are not so many of them. And, okay, so first one is we're going to think about our inclusion bodies, which are just simple organelles for storing some kind of nutrients inside bacteria. So these can either be organic nutrients, so glycogen and polybeta hydroxybutyrate are stores of carbon, organic carbon, whereas cyanophycin is a polymer of arginine and aspartic acid. So it's a store of nitrogen, and carbon. Okay, so these are amino acids. They've got carbon and nitrogen in them. Now, as well as some organic inclusion bodies, some bacteria, or many bacteria, store phosphate. And some bacteria with a metabolism based on sulfur can store up sulfur granules. Now, the basic idea here is that whenever these nutrients are abundant, bacteria will have a tendency to internalize them and store them in the inclusion granule, because then if they run out, they can still live. Okay, they've got some kind of store. Now, some of these granules are directly in contact with the cytoplasm. So glycogen granules and polyphosphate granules, they're just directly in contact with the cytoplasm. Whereas poly-beta hydroxybutyrate are surrounded by a kind of lipid monolayer. Okay, so it's not like a bilayer lipid membrane. Poly-beta hydroxybutyrate is very hydrophobic. So these granules are surrounded by a lipid monolayer with the lipid tail, the hydrophobic tail, which is pushing into the granule of poly-beta hydroxybutyrate. Okay, storage granules help bacteria survive when the food runs out. Another structure inside the bacterial cell is the nucleoid. So this contains the bacterial chromosomal DNA. Now, the nucleoid is composed of DNA condensed with polyamines and proteins called the HU proteins. So polyamines will compact the DNA, and the HU proteins induce curvature in these strands of compacted DNA. And that's absolutely necessary because the length of the DNA molecule, if it was just like strung out without any constraints, is much bigger than the bacterial cell. So this is a bacterium that's been digested with a protease. So all of these HU proteins have been degraded, and you can see the double-stranded DNA is really, really big compared to the bacterial cell. So this condensed form of DNA inside the bacterium is the nucleoid. Now, there are exceptions. So archaeobacteria have different proteins for condensing DNA, and some of them use proteins similar to histones. And planktomycetes, they're really strange because some of the planktomycetes have a membrane-bound nucleoid. So this is like a kind of primitive nucleus that seems to be present in some types of bacteria. This is the kind of thing that is not supposed to exist, right? Bacteria don't have a nucleus. But this guy has got something a little bit like that. Now, another thing that classically was not thought to exist inside bacteria, and by classically I mean about 50 years ago or maybe 40 years ago, the bacteria were thought not to have a cytoskeleton. So nothing inside the cytoplasm that gives some kind of structure to the cell. And that's not true because there are bacterial homologs of the two main cytoskeletal proteins, actin and tubulin. So these MREB and MBL proteins, so MREB is in gram negatives and MBL is the equivalent in gram positives. They are structural homologs of actin. And like actin, they can form filaments. Now, if you look inside this bacterium, Bacillus subtilis, gram-positive bacillus, and it's expressing a fusion protein between MBL and the green fluorescent protein GFP. And this allows you to see the filaments of MBL inside the cells. You can see that they form a kind of helix, and the function of the MREB and MBL proteins is to force the bacterial cell to elongate and give it the form of a rod like a bacillus. Okay, so spherical bacterial cells, cocci, they do not have a gene for MREB or MBL. They've lost it. That's why they're round. Okay, so just this one protein determines whether the bacterium will be bacilli or round cookie. And the bacterial equivalent of tubulin is a protein called FTSZ, which plays an important role in bacterial cell division. So as bacteria grow, the DNA chromosome will start to replicate at the origin of replication, which is shown by this black diamond here. So DNA replication starts from the origin of replication, and you start to get 2 copies of the chromosome produced inside the cell. One copy will migrate, so each copy will migrate to a different end of the bacterial cell, and around the middle, this is where FTSZ starts to polymerize and form a ring-like structure. And this ring is what determines where the bacterial cell is going to split in 2. So the FTSZ itself is not what's doing the work here, pulling the bacterial cells apart, but it's the scaffold which is going to organize all the proteins that are involved in dividing the cell. Okay, so FTSZ playing a very essential role in bacterial cell division and chromosomal segregation. Okay. Now I find that's very interesting because the closest eukaryotic homologue to FTSZ is gamma tubulin. And gamma tubulin in eukaryotic cells is what organizes this thing that I'm drawing here. What's that supposed to be? Yeah, in eukaryotic cells, it's chromosomal segregation. What's the term for this? In French, if you like, and then I'll tell you in English. Huh? Le quoi mitotique? C'est pas le fuseau mitotique plutôt? Yeah, yeah. Fuseau mitotique, the mitotic spindle. Okay. So gamma tubulin is what organizes the mitotic spindle in eukaryotic cells. Okay, so this is kind of an intriguing thing. You know, you think, oh, come on, that can't just be like random chance here. We've got the same kind of protein playing an essential role in chromosomal segregation in bacterial cells and in eukaryotic cells. So the whole process of chromosomal segregation in eukaryotes and in bacteria is very, very different. But somewhere along the line, there must have been a kind of similar process happening. Okay, they must have had a similar precursor. And this FTSZ tubulin protein is somehow at the heart of it. Okay, that's bacterial cytoskeleton. And the last thing I want to talk about for the bacterial structures are endospores. Okay, not endosomes, endospores. So these are highly resistant dormant cells that are produced by some gram-positive rods, like Clostridium or Bacillus. And the function of these spores is to allow the bacterium to survive when conditions for growth are very unfavorable. So they're very resistant to chemical compounds, resistant to, attack by acids, alkali, bleach, detergents, solvents. They can resist high temperatures, and they can remain viable for years or decades. And then at some point, these endospores can germinate and be woken up. So when conditions become favorable again, then these endospores can wake up. So by favorable, that means nutrients are present. Some liquid water is present. Some endospores can be activated by heat, which will allow the nutrients in the water to penetrate into the endospore coat. So how are they so resistant? You have to look a little bit at the structure, which is very, very simple. So going from the inside, in the middle, you've got a kind of tiny bacterial cell with a condensed chromosome, this time associated with these what's called short acid soluble proteins. So if they're acid soluble, that means they're basic, right? And they're basic, they bind to the DNA. There are DNA repair enzymes here, then there's some ribosomes. But there's no metabolic activity, or very, very little going on in the endospore. It's dormant. And it's dehydrated. Then afterwards you have the cortex of the spore, which is formed of a very thick layer of peptidoglycan. And it also includes this small molecule called dipicolinate, which is this thing here. So there's a lot of dipicolinate in the cortex. Then outside you have the spore coat, which is formed of protein and mostly not doing very much. It's just a protective coat of protein. But in amongst there you have some inactive proteases. So this is why the endospore is very, very resistant. Any kind of chemical attack, it just gets absorbed in the protein here. Nothing can get through. So when endospores wake up, what happens? So nutrients are present, liquid water is present, might be some heat. What that means is that some nutrients can start to diffuse through the spore coat and get into the cortex. Once they get there, this will end up, well, will result in releasing the dipicolinate, which will start to diffuse out. And the dipicolinate will activate the inactive proteases in the spore coat and start to break it down. And then that means that the whole spore coat is more permeable to water, and then the core, well, the cell on the inside will rehydrate and start to become active again. Okay, so one classical example of a spore-forming bacteria are Bacillus anthracis, the anthrax bacillum. Spores are formed in animals who are infected and they die, and then they'll release spores into the ground. And these spores can remain infectious for years or decades. That's why if you've got a field that's contaminated, you can get sporadic cases years or decades after the last known case, because these spores can stay there and get transported around in the ground by earthworms, and then cattle can eat them again. Okay, how about spore formation? One of the kind of problems with this endospore idea is you've got these big layers of polymers, peptidoglycan and proteins, that are surrounding a cell that is entirely inactive. So how on earth can this spore produce the cell wall and everything? Because it has no metabolism going on down here. And the answer is, well, it doesn't produce it itself. It's produced by the sporangium cell. And spore formation starts in spore-forming bacilli by an asymmetric cell division, which will form one larger cell, which is going to become the sporangium, and a smaller daughter cell, which is going to become the endospore. Now, the endospore will express these DNA repair enzymes, SASPs, and then shut down its metabolism. But the larger cell, the sporangium cell, will start to engulf the endospore, and then it's going to synthesize all the different layers in the endospore coat. So it's the sporangium cell which is performing all the biosynthetic reactions to produce the endospore coat. And then at the end of the process, these two cells will have very different fates. The endospore will be released, it can survive, and the sporangium cell will undergo programmed cell death and will die. Now, once again, this is very, very interesting because normally when we think about bacteria, we think about cells that will produce two identical daughter cells which are just the same, and then they'll divide again. But here we have one cell dividing into two cell types that have very, very different fates. So there's the basis here of cell differentiation. Now, we all know that to produce a complicated multicellular organism, that's what you need. You need to have some kind of differentiation of cells that are genetically identical into different cell types that do different things. And you also need, during the development of a complex multicellular organism, you need to have some cells that die when they need to, programmed cell death. And so in spore-forming bacteria, these two fundamental processes, they're already there in a very rudimentary way, okay? There's some very simple cell differentiation, and there's some simple programmed cell death. Okay, so that's all we need to know about bacterial cell structures. So I'm not going to read through this, but just this is what I thought were the essential things to try and understand for all of these structures. What's the function, what's the structure, and what's the biosynthetic pathway in the kind of general lines of how bacteria make these different structures. Okay, so that's the end of the first class. So now we're going to go on to the second one. Okay, so here we're going to think about not really bacterial structures anymore, more about how bacteria live, what they need to live, how they can take up nutrients, and then how to grow bacteria. So probably what I'm going to do is do the first 2 points here, maybe start on growth culture media, and then all of this, which I suppose you can call bacterial physiology, will be next week. Okay, nutritional requirements for bacteria. Well, everybody needs a source of carbon to make their biomolecules, and bacteria either require an organic source of carbon or an inorganic source of carbon. So bacteria that require carbohydrates, peptides, fatty acids, some form of organic carbon are called heterotrophs, and bacteria that can mobilize or fix carbon dioxide and then produce all of their organic carbon molecules from carbon dioxide are called autotrophs. Now, yeah, I added this here that, don't get the idea that heterotrophs, they can use anything, okay? So different species of bacteria are specialized in using different types of organic carbon molecule. So almost all bacteria can use glucose as a source of carbon, but maybe not all of them can use cellulose, for example. Okay, so not all of them can use lactose as a source of carbon. Okay, so heterotrophs will be more or less specialized in the use of some particular organic carbon, source of carbon. Another thing that's worth mentioning here are bacteria which can survive only on methane. And I always find it difficult to know whether they should be called heterotrophs or autotrophs. So you could say they're heterotrophs because methane, CH4, it's an organic molecule, right? But on the other hand, it's very, very simple. So these bacteria have got to make a lot of complex carbon molecules out of something that starts with just one carbon, methane. And this is a bit like what the autotrophs are doing. So often, you'll just have them called methanotrophs. Okay, they're kind of somewhere in the middle. Now, this heterotroph autotroph thing, this is related to the nutritional type of bacteria. Oh, I didn't check this out. So, what's going on here? Finally, very, very strange. So in terms of source of carbon. That's what we've just been talking around here, right? So bacteria require organic carbon heterotrophs, can fix inorganic carbon, they're autotrophs. Bacteria also need a source of energy and a source of electrons. So if the source of energy is chemical, they're chemotrophs. If the source is light, they are phototrophs. If the source of electrons is organic, then they are organotrophs. And if the source of electrons is inorganic, they're called lithotrophs. So if you want to fully describe the nutritional type of a bacterium, you need to have a word that has all three of these terms in it, okay? So for example, cyanobacteria, like Anabina, they are photolithoautotrophs. Use light energy, inorganic source of electrons, and fixed carbon. Nitrosobacteria, like Nitrosomonas, they are chemolithoautotrophs. Chemical source of energy, inorganic source of electrons, inorganic source of carbon. And enterobacteria, like Escherichia coli, chemoorganoheterotrophs. Okay, so you saw this last year, so I'm not going to talk any more about this, but it's important concept. Okay, so that's carbon sources. Nitrogen sources, once again, you know, bacteria would love to use organic nitrogen if it's available in amino acids or purine and pyrimidine bases. But often don't have many amino acids floating around in the sea, for example. But you might have inorganic sources of nitrogen, which can either be reduced, ammonium ions, or oxidized in the form of nitrate or nitrite. So pretty much all bacteria can incorporate ammonium ions into amino acids. And a large number of bacteria, most of them, can also incorporate nitrate. First, they'll reduce the nitrate to ammonium, and then use that and incorporate that ammonium into glutamate or glutamine. So for this incorporation, two very frequent reactions are the incorporation of ammonium into, ammonium ions or ammonia into alpha ketoglutarate, just with one reaction to produce glutamate. So you've got this ketone group here, which is replaced by the amine group from the ammonium. So one simple reaction or step, and that's it. You've got organic, you've incorporated this ammonium into an amino acid. So that's when ammonium is at high concentrations. If ammonium is at a low concentration, then bacteria will tend to use another enzyme, which will incorporate ammonium into glutamate. So from glutamate, then this ammonium will be incorporated into glutamine. And this time you need to use ATP as the coenzyme. So both of these reactions require some kind of metabolic energy. This one requires NADPH and this one ATP. But, okay, it's only costing you either one ATP molecule or one molecule of NADPH to get the inorganic nitrogen into an amino acid. And that's much easier than nitrogen fixation of nitrogen gas. So only very few bacteria can do this. So rhizobium and cyanobacteria. And this is a very costly process. So this is 16 ATP and 4 molecules of NADPH just to get one molecule of nitrogen gas incorporated into ammonium. Okay, so almost all bacteria can incorporate ammonium or nitrate. Very, very few bacteria can perform nitrogen fixation. Okay, organic molecules also have phosphorus and sulfur. So phosphorus essential for nucleic acids, right? And sulfur essential for cysteine, the amino acid, and also some important enzyme cofactors. So phosphorus, well, if it's present in phospholipids, DNA, RNA, this can be incorporated by bacteria, and all bacteria can incorporate inorganic phosphate. Sulfur can be present if amino acids are around. Bacteria will use the sulfur in cysteine or methionine. But if no organic sulfur is around, then most bacteria can use hydrogen sulfide, which can be easily incorporated into cysteine with a simple reaction from O-acetylcysteine together with hydrogen sulfide. This will give you the amino acid cysteine and kick out acetate as a side product. So once again, one simple reaction here. Okay, so all bacteria need a source of some source of phosphorus, sulfur, nitrogen, carbon, and they also need mineral salts, sodium, magnesium chloride, potassium, just to maintain the ionic balance inside the cytoplasm. So they need reasonably large quantities of these ions, and they need trace quantities of other metal ions. Manganese, cobalt, which are going to be cofactors for some enzymes, and all bacteria require iron because this is going to be very important for any proteins involved in electron transport. And finally, some bacteria may require growth factors, vitamins, amino acids, or purine and pyrimidine bases. So a growth factor is something which is essential for bacterial growth, but the bacterium cannot produce it on its own. Okay? So bacteria which do not require growth factors, they can make all of this stuff themselves. They are called prototrophs. Bacteria that require one or more growth factors are auxotroph for something. Okay? So if the bacterium requires thiamine, it can't produce it, then it will be auxotroph for thiamine. And then you need to add this to the culture medium, otherwise they won't be able to grow. Okay, so time for me to give you a question here, just to test this. I don't know if that will actually work, this link here, because it should be the different one. Let me just check here. Is that link working? Yeah, it's okay? Okay. It works or not? No, it's not working, right? That's because it's the old link from last year. I was pretty sure I changed these. Okay, this should work. Oh. I got one vote so far. 2 votes. Okay, what do we got? Most people got the right answer. Okay, photolithoautotroph, autotroph for biotin. So why is that the correct answer? Okay, so we've got a medium. It only has mineral salts. There's no organic carbon. So it's got to be a lithoautotroph, no organic carbon. And it needs light, so it's definitely a photolithoautotroph. So it's one of the first three. No, it's either this one or that one, photolithoautotroph. And the question is prototroph or auxotroph for biotin. Well, if it needs biotin, then it's auxotroph. Okay, that's what it means. That's what it means. You need biotin, you're auxotroph for biotin. And if you understand that, then you have no problems in understanding bacterial growth requirements. Okay, so we know what bacteria needs. How do they capture, how do they take up the different things that they need? Well, now it's just the same kind of question as for any type of cell. The bacteria have to be able to get things across the membrane, either specifically the plasma membrane. So one mechanism is passive diffusion, but that will only work for small gases like oxygen, carbon dioxide, methane. They can freely pass through the bacterial membranes. This is my automatic translation. It's terrible. I didn't check this. Passive diffusion. This is supposed to be facilitated diffusion here. And like I said, just check. No, it was translated by the OMIST tools. You can upload your slides and you can translate them into any language. It's on like the university server. But you have to check them because for technical terms, sometimes it's not always correct. So facilitated diffusion, okay? You have there's got to be some kind of pore in the membrane to allow molecules to cross it, but there's no energy input. So that means that molecules will only follow the concentration gradient, okay? And for facilitated diffusion, there's a difference between permeases and porins. So permeases, they're specific for a particular molecule, whereas porins, they're just like a hole with a particular diameter, and they will let through any type of molecule of a particular size. Now, these are essential in gram-negative bacteria, right? So in gram-negatives, you've got an outer membrane, peptidoglycan, and you have the plasma membrane. You need porins and permeases in the outer membrane, and they will just allow molecules to pass following a concentration gradient. Gram-positives, they don't need to worry about that because they've just got the peptidoglycan layer, which is permeable to small molecules. Like a molecule of glucose or an amino acid can just diffuse through peptidoglycan. No problem. Now, so if you've got a molecule of glucose coming into a gram-negative bacterium, it's going to go through a porin or a glucose permease, and it's going to end up here in the periplasm. But in order to keep this concentration gradient, bacteria will have to import this glucose from the periplasm very quickly into the cytoplasm, so that the glucose concentration stays very low here, right? And then in that case, more glucose will keep coming in. And for this part, this step here, all bacteria need to use active transport, because the concentration of whatever you want to get is going to be higher inside the cell than in the periplasm, because you want to keep the concentration low here so that the flow can keep coming in. Okay, so there are three types of transporters used by bacteria: the ABC cassette transporters, proton or sodium symport antiport systems, and what's called group transport. So we're going to look very quickly at these. Oh, and then finally we'll talk about, how bacteria get iron and other metal ions. So it's also a very important system. So ABC transporters. So ABC is for ATP binding cassette, and this is telling you that the energy for the active transport comes from the hydrolysis of ATP. And there are two components in an ABC transporter. There's a solute binding protein, which is in the periplasm. So this solute binding protein will bind whatever wants to be transported. For example, phosphate, some amino acids. And then the solute binding protein, together with its cargo, binds to the pore. And then the ATP hydrolysis will pull off whatever the solute is and bring it into the cytosol. But you're going to spend, for example, 1 molecule of ATP for every molecule that you import into the bacterial cell. This is the most expensive way to import something. Okay, right, so phosphate, amino acids, and some sugars like maltose, galactose, ribose in E. coli. Second transport system are proton or sodium symport or antiport systems. So once again, you have the periplasm here and the cytoplasm here. So some bacteria can constitute a sodium gradient across the membrane and then will transport sugar together with a sodium molecule. So in E. coli, but most of the time, this is going to work just simply with proton symport. So the proton is transported across the membrane at the same time as the solute. And as we saw, bacteria maintain a higher concentration of protons in the periplasm than in the cytoplasm. So the proton is moving down the concentration gradient. That's energetically favorable. And this is what pays for the import of the solute, whatever it is, which is going against the concentration gradient. So proton symport in E. coli is often used for amino acids or these 4-carbon molecules like malate and oxaloacetate. So they've got a carboxylate group. It kind of makes sense that they go together with a proton. So this should be kind of easy to remember which one is transported by proton symport. So this is slightly more economical than ABC cassette transporters. It costs you 1 proton in your protein gradient to import something. Not bad. And the most efficient import transport systems are the group transport, also known as phosphoenolpyruvate sugar phosphotransferase systems. Now, what is happening here is, what is happening and who uses them? So basically, this is going to be important for importing sugars, particularly glucose, and especially in anaerobes, obligate anaerobes, and many facultative anaerobes. Aerobic bacteria generally don't use this system for importing glucose. So how does it work? Okay, well, the big special thing about phosphotransferase systems is that the import is coupled to the phosphorylation of the glucose or the mannitol. So, and the energy for this, where does this come from? Well, it comes from the cleavage of a phosphodiester bond here on this protein that's part of the pore complex, protein 2B, which is phosphorylated. So the cleavage of this phosphate bond here will provide the energy to import the glucose and phosphorylate it at the same time. Okay, so where does this phosphate group come from? It comes from a series of transfers from other proteins in the system, but way back at the beginning, the origin of this phosphate group here comes from phosphoenol pyruvate. So this is phosphoenol pyruvate up here. And as this phosphate group is passed from phosphoenol pyruvate to the enzyme 1 protein here, this will release pyruvate. Now, I don't know if you've seen this in biochemistry yet, but this is the last step in glycolysis, phosphoenol pyruvate to pyruvate. And normally, this is coupled to the synthesis of one molecule of ATP. So normally this phosphate here would be combined with ADP and give you one ATP. So using this phosphate in the PTS system, it's the same as using one molecule of ATP, okay, in energy terms for the cell. But the clever thing about it is that this cost of one ATP, it does two things at the same time. It has the transport of the glucose across the membrane and its phosphorylation into glucose 6-phosphate. That's important because the first step in glycolysis is the phosphorylation of glucose to glucose 6-phosphate. So if you were importing glucose by an ATP binding cassette transporter, it would cost the bacterial cell 1 ATP to transport the glucose, then 1 ATP to phosphorylate the glucose into glucose 6-phosphate. You spend 2 ATP before you even start to metabolize. Whereas here, with this system, you only spend one molecule of ATP. And that's very useful for bacteria. It's economic, it's not wasteful. And it's more important for anaerobes, because anaerobes will probably be generating their energy by fermentation, which is not very efficient. So every molecule of ATP is really important to save. Whereas for aerobes, they're performing respiration, they get loads of ATP per glucose molecule. They're rich. Oh, 2 ATP, take it, my man. Okay, so this is why group transport and the phosphotransferase systems are really clever and important, especially for anaerobes. Oh yeah, that's why I said this. I just said that, okay. Right, last thing to think about importing is iron, and it's also true for other oligoelements. So all bacteria need a reasonable amount of iron, but in most environments where any oxygen is present, all the iron is going to precipitate out. So the concentration that is available in a soluble form is very, very low. So they have to be able to pick up iron even though the concentration is very, very low. So the way that many bacteria do this, including E. coli, is to secrete a molecule called a siderophore. And the siderophore here, which is called enterobactin from enterobacteria, can chelate iron at a very high affinity and will form a very, very stable complex which can pick up iron at a really tiny concentration. And then all of this complex is re-imported into the bacterium by an ABC carrier system. Siderophores really essential for bacteria to pick up iron. and often other metal ions that are essential oligoelements. Okay, so I've done transport systems. We've got about 10 minutes, so I think we can just think a little bit, start thinking about culture media. So we know what the nutritional requirements are for bacteria. We know how bacteria take up nutrients. So what kind of media, what kind of things do we need to put into a culture medium for bacteria to grow? Okay, well, one type of medium that you can use is something called a defined or a synthetic medium. And in a defined medium, every single component that's in the culture medium is known precisely. So this is one example of a defined medium. Glucose, source of organic carbon, sodium hydrogen phosphate, potassium dihydrogen phosphate, ammonium sulfate, magnesium sulfate, calcium chloride, iron sulfate. So you've got a source of organic carbon, you've got phosphorus, you've got nitrogen, you've got sulfur. But it doesn't include any growth factors. So this is what's called a minimal medium. So only bacteria that are prototroph for all vitamins, amino acids, and nucleotide bases can grow on a minimal medium. They can make it all out of carbon and these inorganic sources of nitrogen and sulfur. So this is useful to study the nutritional requirements of bacteria. So if you've got a bacterium, try and put it in there. If it grows, you know that it's prototroph. If it doesn't grow, well, you can try adding growth factors. Okay, you can try adding vitamins, amino acids. Start by putting them all in, and then take one out after the other, and then you can figure out what things it can produce and which ones it can't produce. Now, in theory, in theory, if you look at this composition of the medium, it shouldn't actually work because something is missing. What's missing here compared to what I said before about bacterial growth requirements? Even prototrophic bacteria should not be able to grow on this. That's missing, but if they're prototroph, they can make them. So they won't need to add the growth factors. But even if it can make all the growth factors, there's something that in theory is missing. Carbon, organic carbon, we got it. Nitrogen, sulfur, phosphorus, we got it. We can, yeah. No, we got a source of carbon. If bacteria are organoheterotrophs, they can grow on this, right? They don't need light. What's the other thing that is supposed to be an essential for bacterial nutritional requirement? It was on the slide. Okay, hang on. Let's, where's my pen here? Okay, somebody give me a letter. What? Huh? Huh? M. Huh? Yeah. Okay, so oligoelements. What did I say for oligoelements? Trace elements like cobalt, manganese, this kind of stuff. They aren't in the composition here, right? So how come bacteria can grow with this? It's because if you don't use a very, very pure form of these chemicals, you'll have a little tiny trace of other ions, metal ions, as a trace contaminant in the sodium hydrogen phosphate. And because you've got 16 grams per liter of this, the tiny amount that you have as a contaminant is enough. So that means that if you want to study the requirement of bacteria for oligoelements, you have to order the purest kind of chemicals that you can buy. But if you just want a minimal medium, doesn't cost too much, just get the cheap one and there will be enough oligoelements. Okay, that's not really massively important, but I think it's interesting anyway. So last thing that we're going to do today is to, another translation mistake here. Okay, is what's called a complex medium. So in a complex medium, you don't precisely know all of the constituents of the culture medium. And so a typical example of a complex medium is LB medium, called lysogeny broth. So here you've just got 3 ingredients. You put in some yeast extract and peptones. So peptones are a hydrolysate of something which is rich in proteins. Could be meat or soya or casein. So complex media, they're often very easy to prepare. You've just got to weigh out 3 things and then put it in a liter of water, shake it up, and you're ready to go after you sterilize it. But it's called complex because we don't really know what is in here. This is a complex mixture of compounds. Yeast extract, it's a complex mixture. So here in the peptones, you have all the amino acids that you need. You'll have a source of carbon to grow on. Yeast extract will have all the vitamins and purine and pyrimidine bases. So these 2 components, all the potential growth factors that bacteria may need will already be there. Okay, so a complex medium is just useful to allow you to start cultivating a wide range of chemoheterotrophs without really knowing what they need precisely, okay? So not all bacteria will grow on this, but as long as you don't have any specific particular nutritional requirement, many bacteria can grow on a complex medium like this. So I'll stop there. Next microbe of the week. What about Sunday, 7 p.m., 5 p.m., something like that? What do you think? So the next microbe of the week question, when do you want me to send it out? 5 p.m. Sunday. Okay, I'll do that. Okay, well, enjoy your weekend, everybody.